All right, welcome back. The stragglers are coming in. I'm going to go ahead and get started with introducing our last speaker of this session is Angela Gutchess. We're very excited to have her here.
Sorry. Okay, so I'll be talking about some of my lab's recent research on culture. As I try to figure this out, I should also give basically co-authorship to this talk to Joshua Go. Many of you know Josh is also an alum from Denise's lab. If there were not still quarantines in Taiwan, he would probably be here giving his own talk, but for today I will speak for him in ways he may or may not agree with. But I'll say a little bit at the end about his line of this work. Much of what I'm presenting today, though, is collaborative. So even though Denise published this paper in 1999 with Trey and Dick, really laying out reasoning for why to look at aging across cultures, there's been relatively little work in this space. So since this paper, she had other reviews, some of which I was lucky enough to be a part of, looking at more of a cultural neuroscience perspective on age and culture. And then she was also part of what is probably the first cognitive neuroscience study of cognitive processes across age and culture. But this was 2007. So since then, there's really continued to be a lack of aging research across cultures, particularly from a neuroscience perspective. So this is part of my first hedge of why much of what I'm going to be talking about today, well, really all the new data I'm showing you is about young adults across cultures, and I'll talk about where we're going to go with this or where we're going now with aging and some of the frameworks we're using to think about this in ongoing data collection. I'm going to be talking about work we're doing in terms of looking at different stages of memory and how culture could influence that. The other final caveat before I get started is I think Naftali had a line during his talk about the findings don't always follow the theory or aren't as straightforward. And I think it's fair to acknowledge that up front, that I feel like the behavioral work we've done has come out in more systematic ways that follow from the theories. The neuroscience data, not necessarily. So I'm hoping at this point in the afternoon, you're all open to more speculation and we can talk about where this could go and not find that frustrating. So first, a little bit about, enough caveats. So first, why study cognitive neuroscience across cultures? As many of you know, much of the psychological research has primarily been conducted in Western cultures. This has been dubbed the weird problem, that we're mostly studying Western-educated people from industrialized, rich, and democratic societies. And some analyses say that this is 96% of subjects from Western industrialized countries in mostly behavioral psychological journal articles, and 68% of the subjects from the United States alone. So clearly we're not really studying, humanity, we're studying a narrow slice of humanity. So that's why doing this type of work is so important, to think about first, doing the broader sampling necessary to have accurate models of cognition and understand those universal properties across subgroups and what differs across different groups, what's truly a universal and what is not and that we've assumed is so. And this is particularly true also of aging and neuroscience data, that much of the work is happening in the West in a limited set of countries. So focusing on how culture might influence memory, the frameworks we've started with were really influenced from the time at Michigan by Nick Nisbet. And his idea is that culture might really shape which cognitive processes and styles of information processing are prioritized and used predominantly in one group compared to another. And we thought about how this might shape memory specifically, long-term memory. So at some level, it may be similar to social research, suggesting that memory is a way of picking up what a culture values and prioritizes. It's important to you. It's about your orientation in the world. You're going to see the world that way and remember it that way. And Chi Wang has done a lot of nice work with autobiographical memory, mostly really all behavioral, looking at how what we choose to tell or choose to store in terms of our own narratives about ourselves and what happens to us. We've thought about extensive experience in a culture affecting a number of different types of information processing. One is the type of knowledge and strategies you acquire. So maybe that's actually taught in school, how you learn to do arithmetic, or in terms of memory, what you use when doing an outline. It's directing attention in a complex environment. We're always in environments where we can't take in everything. How is some information selected for further processing at the expense of others? And in turn, how is that information encoded into memory versus forgotten or distorted in systematic ways? So what my lab's been looking at in recent years is focused on this idea of memory specificity, that we can remember information in a number of ways and different levels of abstraction or detail. In contrast to much of the research that came before, like with autobiographical memory, we've tried to really standardize those encoding experiences so that people are studying pictures of items, like the ice cream cone on the left, and later on they're going to be tested about, do they remember the perceptual details of that object? So can they later on recognize that, yes, I saw this item before on the left, it's the same one I saw before? Or if they get this ice cream cone on the right, and they should call it similar in some of the paradigms or new, can they distinguish that from just knowing they saw an ice cream cone before? So we're giving a standardized encoding experience of seeing these pictures and then testing memory in different ways across cultures. So what we'd predicted, and what I'm going to show you, is that, again, you heard some of this in Dick's talk, that the more analytic processing style that's expected for Americans might support memory for these details, of these perceptual aspects of pictures, versus a more holistic style might not support that memory for detail. And that would be a more East Asian style. In terms of the memory specificity differences we found behaviorally now across a number of studies, what we tend to find is for the specific memory, distinguishing the one ice cream cone that you studied from a different exemplar of an ice cream cone, Americans are having some advantage in memory for the specific items compared to East Asians. Whereas in general memory, just knowing I saw an ice cream cone or not, regardless of the details, we actually don't see cultural differences. And we've seen this in a number of studies now, some with backgrounds, which is important for thinking about holistic processing, some with more signal detection and analytic approaches, and yes, no, that helps with some of the details of analyses I'm not going to talk about today. But this has been a robust finding, that we're finding these behavioral differences across cultures in remembering the items in detail. So to set up the fMRI data I'm going to talk about today, I'm going to really give you broad brush strokes about some of the methods. So one data set is from Encoding. This was collected a few years ago, so sample sizes are a little smaller. It was run entirely in the United States, and what that meant was that the East Asian participants had moved to the US on average 2.5 years ago, typically for college or graduate school. They're mostly from China, but it could be a variety of East Asian countries. We normed the stimuli some, but in the more recent work that I'll be showing you, that we looked at rest after an encoding period and then retrieval, we had more systematic norming of older adults in Taiwan and the US to really make sure the stimuli were age and culture appropriate. We also are using the items in the mnemonic similarity task that I think many of you have used and is commonly used in the literature. It's a slight variation of what we used before. Here we're collecting data in the US and Taiwan. Our sample sizes vary a bit. We're still collecting data for the Americans. Time any samples pretty much set. So the first number is the N that I'm going to have for the rest analyses I'm showing you, and the second number is the N for the retrieval analyses that are, these are a little behind what we just did for the rest analyses. And again, all young adults at this point. So in terms of the encoding of specific memories, what we're looking at here is how do the regions and processes that are associated with successfully encoding specific memories differ across cultures. So when you're first encountering this information and forming memories, what brain regions are being recruited that help you to form those really specific detailed memories compared to when you don't need that same specificity or level of detail? And prior work had really implicated medial temporal lobe regions in the formation of specific memories. And we predicted that Americans would recruit those regions more than East Asians, given the behavioral benefits for Americans of forming memories from more specific objects than East Asians. And this first study was led by Dr. Laura Page, who graduated from my lab a few years ago. So what I'm going to show you in the analyses are the difference between the correct same items, so forming those detailed memories, compared to when you form the memories for the similar items that, again, don't require as much detail, to just be like, I saw an ice cream cone, was it the same or not? I don't know, I'm going to call it similar, whereas really being precise with that correct use of similar. And the pattern I'm actually going to show you, I just predicted Americans greater than East Asians, the pattern we found is the opposite of that. So in a number of medial temporal regions, including fusiform, perihippocampoid gyrus, and hippocampus, all in the left hemisphere, we're actually seeing regions that East Asians are recruiting more than Americans for the same comparison to similar. So the orange bar on the left for same is going to be higher for the East Asians than the blue bar for Americans, and that difference is either negligible or even maybe potentially in the opposite direction across cultures for similar. So again, regions supporting the formation of the detailed memories. So basically, we're seeing that East Asians are recruiting more of these medial temporal regions for specific memory formation than Americans. And again, it's surprising we'd predicted the opposite. We did some follow-up analyses trying to look at difficulty. None of the metrics were really perfect for this, familiarity of items. Nothing seemed to explain, why we were getting this pattern. We thought there's some potential that the greater engagement of regions may reflect the greater demand to form specific memories in East Asians. So again, we're selecting those items that was formed in detailed memory to get the correct same response. Maybe to do that just requires some additional engagement of regions to form those memories in the culture where that should be a less practiced or preferred processing style. The other thing we thought going forward is thinking about this in terms of, well, what are the kind of trade-offs or different processes across different stages of memory? Encoding is only one piece of what's ultimately leading you to retrieve information. So the next couple studies I'm going to talk about how cultures are varying at these different stages. So we next, in a new data set, what we've analyzed, the original focus was on retrieval, but during the pandemic, we thought we could analyze what we have so far at rest. And we framed this around post-encoding rest. We did the same type of encoding task as what I just showed you. People saw a number of different items. The memory was tested later on. So basically, so we might find cultural differences in what's happening post-encoding. And you may think of this as reflecting memory processes. There's a lot of really interesting work, initially in rodents, now in humans, suggesting these kind of downtimes of rest periods, people may be spontaneously forming memories. So in the case of rodents, you can see them kind of tracing their memory path through the pattern of neurons that are engaged. In humans, you're not seeing it with that precision, but it may be that the prior experience of seeing information with a lot of perceptual details is going to shape what's happening in this period that may help with consolidation of memories. It also, though, may reflect more general processes, potentially more trait-like differences across cultures. So doing more holistic or analytic processing that might also be happening during the spontaneous period of, basically we're going to be looking for what regions are engaged with other networks. So it might pick up these general differences in how older adults and young adults think or approach information, even when they're kind of in downtime. So the caveat is that the current design only has one resting state block, encode, rest, so we can't distinguish these two explanations, and I'll talk about that with the interpretation as well. So we conducted resting state functional connectivity analyses using those seed regions from the prior encoding study. So those regions that showed the cultural differences in medial temporal regions, we used those then to look at how do cultures differ in the regions that are connected with those medial temporal regions during rest. And those basically came from hippocampus in green, perihippocampal gyrus blue, and red. Blue's anterior, red's posterior. So these were our seeds to then look at connectivity with these regions and how that differed across cultures. By focusing on medial temporal regions, we wanted to kind of maximize the chances that we were looking at memory-related processes. But again, caveat with that we don't have a direct comparison of after encoding versus not after encoding. And this work is led by a current PhD student, although about to graduate, Wanbing Zhang in my lab. So what we found that's most of interest for what I'm going to show today is we found differences in connectivity with the left anterior perihippocampal gyrus seed. So again, this region came out of the encoding analyses. We're now looking at a different data set, different subjects of how those regions were connected differently with other brain regions. So what I'm showing you here is there was a set of regions that kind of corresponded to ventral stream in both left and right lobes, including temporal pole, middle and inferior temporal gyri. The East Asians had stronger connectivity than Americans. So the gray bars are stronger connectivity with that left anterior perihippocampal region than the Americans in black. So it suggests some cultural differences. To go or in? Oh, okay. All right. Don't worry, I'll try to not speed up. It's also these group differences were also substantiated with individual difference scores on some of these measures. So participants outside of the scanner completed scores of independence versus interdependence. What we found is the subjects who were lower on independence, had higher functional connectivity. So this would be in line with what would be expected to be a more East Asian style of being less independent, and that really corresponds to the pattern we're seeing, that the stronger connectivity for East Asians fits with lower levels of independence on that individual difference comparison. In contrast to the regions that were more connected for East Asians than Americans, we found a different set of regions that were more strongly connected for Americans than East Asians. And they tended to be more dorsal stream, particularly frontal lobe. And again, the individual difference values map onto that. So that same independence measure, now higher levels of independence, the more American style, is linked to stronger functional connectivity. We also had measures of cultural values, including tradition, which would be a more East Asian style, to be high on that score. So again, the lower tradition being associated with the higher functional connectivity, again, would fit with the pattern of group differences that we're seeing. So to summarize those findings, we're seeing these differences potentially with East Asians engaging more kind of ventral stream regions connected to this anterior perihippocampoid gyrus seed, Americans more dorsal, also connected with those self-construal and tradition measures. Part of this could reflect the consolidation of weaker memory traces. So I mentioned that could be memory processes, after an encoding task during rest. It actually seems like the weaker traces are what might be, the weaker memories may be what's getting consolidated more or showing more functional connectivity during this stage. So what you see in those functional connectivities is the more American style would actually be what Americans are worse at compared to the East Asian style and the functional connectivity with regions of what they're worse at. But it also could be more general processing biases across cultures. So again, dorsal ventral stream with what versus where. Other literature mapping onto this is this anterior temporal versus posterior medial system of conceptual versus contextual remembering, or more gist versus detail networks. So future directions would really go after this by manipulating processes to really manipulate what's happening before this rest period, or even looking during task versus rest to see do the same patterns really emerge when you're doing these actual tasks. Looking at baseline versus post-encoding rest. In terms of aging, this may become interesting because one of the potential interpretations is this more gist system that tends to be more anterior versus more detail processing. And we know that with age, people seem to shift more, at least based on Western data, to a more gist-based processing. So we may find in older adults that there's this difference in terms of what pattern is more predominant or whether these cultural differences are really persisting with age. In terms of retrieval for specific memory, so we thought about this in terms of pattern separation completion framework, that the medial temporal lobes and hippocampus, specific regions seem to care about pattern separation, so forming a new pattern to represent a new exemplar or concept versus completing a kind of familiar or known pattern. So what that tends to look like is when you have really high similarity items like these two umbrellas that are very similar but differ slightly in perceptual features, that you need you need to really engage pattern separation to be able to distinguish a new exemplar that's so similar from an old one. So we thought about this across cultures that potentially it's not going to be a limitation in the ability to use the hippocampus or do these pattern separation and completion functions, but potentially differences in the bias of how readily you engage in pattern separation versus pattern completion. And this work is led by Kristen Legere, who's a PhD student in my lab. So what we've seen for the retrieval data is, first of all, replicating the behavioral pattern we've seen before, with Americans having better memory for those detailed pictures versus similar pictures for the Americans in blue compared to the Taiwanese in red. I'm plotting this differently than I showed you in the first behavioral slide because it's also binned by the Yasun-Stark bins of which items are easier versus harder. So there are 5 different bins based on predominantly American or all American, but potentially Asian Americans are in that sample as well, of which items are the easiest versus hardest. And you actually see that the cross-cultural differences are larger for the easier items, which is not what I would have expected. So it seems that Americans are coding that additional detail for the items that are more distinct from each other. It's helping them more later on than for the Taiwanese. So first, replicating the behavioral pattern in this sample at retrieval. What I'm showing you in terms of neuroimaging data are for similar items, when people correctly reject and say that, no, that's not the item I saw before, even though it might be similar, compared to when they actually make that mistake and say, oh, that is the umbrella I saw before when it wasn't. So you see actually this left inferior frontal region that's differing across cultures, with Americans engaging the region more than East Asians. For correct rejections versus false alarms. And I predicted these medial temporal differences related to pattern separation completion. We're not seeing that on a first pass in young adults. We need to do some region of interest analyses when we have the full sample. But this has got us thinking about future directions related to retrieval monitoring. That it may be some of these more strategic processes than we would have predicted that are potentially contributing to cultural differences and rejecting similar information compared to when you make that error. So to summarize the imaging data I've showed you so far, at the time of encoding, we saw some differences in medial temporal regions that East Asians engaged more than Americans. At rest, there may be cultural differences and potentially the interplay or trade-off between dorsal and ventral or anterior and posterior memory systems that could be related to the consolidation of weaker memory traces, but also just may reflect more general processing styles. And at retrieval, we see this difference across cultures with Americans engaging left inferior frontal gyrus more than East Asians when they're rejecting similar lures. And we're designing some behavioral studies now to really go after, does this reflect cultural differences in retrieval monitoring, which isn't something that literature has looked at so far. So the other thing to think about that future analyses should be able to look at is whether there are trade-offs across the memory stages. That it's interesting that East Asians show more engagement of regions at encoding, but Americans more engagement of regions at retrieval. That there may be some interplay between how you process information, how well you encode the information, versus what you need to do later on to resolve interference. So I think that will also be really interesting to look across those stages of potentially which culture group is weighting which in terms of processing. And again, the need for iterative behavioral studies that I've suggested some interpretations that we then need to really go back into the lab and design studies to directly test those explanations. In terms of the age difference and what we're looking at now, again, that pattern separation framework that we started with for the retrieval data is really stemming from the aging literature, which a number of studies have suggested that older adults have more difficulty with pattern separation than younger adults, and that's linked to decreased engagement of medial temporal regions. So patterns like this, that for older adults in the gray bars compared to the black bars of young, you're seeing age differences in these different pattern separation types of tasks. So in terms of how we thought about this for a broader framework to study across cultures is whether these patterns are culture invariant. So it may just be replicating the work that's been done so far to compare across cultures and say, are these age differences really age differences or are they culturally specific age differences? My prediction is actually that we will get convergence with age. I'll say a little bit more about that. So it may be that the initial advantage Americans have on this task is lost by the time they get to be older adults. We're also conducting for my lab some of the first longitudinal work if we ever get that first set of data collection done with COVID, but we'll be bringing the subjects back in to start looking at predictors of decline across cultures, really influenced by the ideas many of you have talked about already, that's starting to look at who's declining more or less within a couple of years, and is that really consistent across cultures, or are there distinct processing differences or trajectories or protective factors? So the way we've thought about this is in terms of, again, kind of a loose framework for thinking about this, but in terms of the kind of purple regions that are more temporal parietal network, that these regions tend to be affected by both aging and Alzheimer's disease and have effects on processes, including memory. So that's a framework based on what I've been talking about today, but we thought about in terms of culture that maybe the vulnerability of this set of regions to pathological aging processes may mean that culture exerts less flexible effects across the lifespan or in older adults. So that would lead to the prediction I mentioned that we may get convergence in these memory processes in the older adults across cultures. Whereas these green regions, a more frontostriatal network, also affected by aging, but less so by pathological aging like Alzheimer's disease. And these regions have really mediate a lot of decision-making and value-based decision-making tasks. So it may be that culture can continue to have malleable effects on the engagement of this frontoparietal network over the lifespan, given that it's not as maybe fixed in terms of pathology. And the regions in green and the decision-making tasks are really the work Joshua Go is leading up. So we'll be able to look across these two systems and processes in younger and older adults soon. So just to conclude, I want to mention that the idea of studying aging and memory across cultures is, again, testing for universal patterns of cognitive decline, risk factors, protective strategies. And one of the promises that we haven't seen so far with aging research across cultures is thinking of it as a naturalistic experiment. You have these lifelong experiences with different cognitive strategies, information processing biases, diet, social context. It may be that some of the outcomes are better for some cultural groups than others, and finding those kind of critical ingredients that then you can bring back to more controlled intervention studies and apply more broadly. So I want to conclude by acknowledging collaborators on this work I presented today, and especially the graduate students who led the work, funding source from NIH. And I also wanted to, since I talked about Denise last night, I was doing less of that in my talk today, but acknowledge the really important collaborations I've made during my time in the Park Lab that have continued past it. So that's been really critical for the cross-cultural work on the top row, but even extends to some of the Americans I got to work with after my time in the lab. So really great experience, so thank you all for that.
I think we're going to move straight into the discussion part. So I'm going to bring up Dr. Kristen Kennedy. Here you go. Thank you.
All right. That was a great set of talks for this session. Also very eclectic, so I'm going to relieve some of your working memory burden and remind you what we've just heard. So we've heard about dedifferentiation and GABA and inhibition. We've heard about wisdom, and maybe we're headed toward more wisdom, or maybe we're not. We've heard about interesting interventions, and older adults like avoiding sharks. And we've heard about culture and how it may and may not shape the brain in ways that we expect or don't expect. So first, I think we should see who has questions for Angela, since she did. Yeah. There are microphones somewhere.
All right. Hey, great talk. That was I love that you're enlarging the tent, right, and giving us a deeper sense of the generalizability of some of the things that we think to be, relative truths. I guess, so I like that the approach starts with using paradigms and stimuli that we have a sense of. But I guess the one thing I was wondering is, to what extent do you think any of the inferred differences in the processes that you're seeing are a consequence of the stimuli, right? So one thing I noticed is that, it's not necessarily the case that the stimuli are normed across the cultures. And there are differences in objects that cultures are most familiar with. So as one example, I know if I was to try to discriminate between two cricket bats, I'd have a harder time than discriminating between two baseball bats, even though I'm from India. So I'm just wondering, I can imagine that would be the next step, right? To see whether or not norming and identifying objects that are equally relevant would lead to the same differences.
Yeah, that's a great question, and one that comes up a lot for this work, as you can imagine. So for the newer data sets, we did try to do better norming. The MST task has, I think, 500 some images. So we had those through Qualtrics, older Taiwanese, older Americans. One of the things I learned from Denise's lab, there were some early norming studies I was involved with, and just rating on a 5-point scale, we realized was problematic. We'd have older adults in that older study give a 5 that they were super familiar, and then they couldn't name it. So one of the things we've tried to do, and that may be partially trying to save face if there are different stigma about Alzheimer's or not knowing things. So we tried to make sure it was ratings as well as naming. So we threw out items people couldn't name, and I forget what the cutoff was for familiarity. So I feel the other piece of it too was having a bicultural student in the lab go through and really eliminate things that just either had text on it or just seemed not good. That's different than an older adult, from Taiwan. This was a student from mainland China. So I feel better about these, but I think in general, I think the real answer to your question is moving to different stimuli, abstract stimuli, would be a really important next step. And we're doing some of that behaviorally. I'm not sure if it's going to come out the same, but again, one of the things I think from talking to Dick years ago is just the different types of, all abstract stimuli are not equal. That it may be some of these perceptual properties, and we're also doing some work on spatial frequency, that also matter across cultures. So yeah, I've tried to move to visual items instead of language-based items, but there's still language involved. So I think the short answer is just more work needed by lots of people approaching this in different ways.
Thanks, Angela. So yeah, great talk, and I guess my inhibitions are down because I might actually have, I feel a little like Dave Meyer because I think I have 3 questions, but so you're ready. So the first one's just a technical one. So on your encoding study, did you actually take a look at memory performance? Because I think it would be very likely that if the memory performance was down, that could really explain a lot of the effects. The other one, so I actually wonder, how do they do, going back to THADS, is this another way to have less distinct representations? And then the third is, I'll just make a suggestion, on the resting state follow-up studies, I actually think it would be incredibly cool for you to collaborate with Ken Paller, and see if during sleep do you get these different replay patterns, and do we either lose some of these cultural differences or do they actually get accentuated? So you can pick whichever one of those you like.
Well, working memory dictates the last one. But yeah, I've also thought, I mean, maybe not in that exact way. I think that'd be a super cool study. I even just thought having longer consolidation periods, like given all the sleep research, these things aren't happening in 30 minutes, and it may be the differences are exaggerated or lost, with a longer period of time. Let's see, what were the other ones? I know I had an answer to something. Yeah, so the question about what is de-differentiated or less specific, I think that is the question, because some of the data that I didn't talk about but I showed is actually the effects have moved around a little bit in some of our studies. That in the more recent ones where we've used a yes/no paradigm to have better signal detection comparisons, it actually looks like it's when the old items are in the comparisons that Americans get their advantage. So it may not be resolving interference, it may be more how much detail is represented in the information that's there. But yes, how is it that it's represented and what's lost, what's gained or filled in? And that's where some of the false memory work's been nice to see how things are distorted. And behavioral, yes. So in the encoding study, we did not get the memory effect. And I think we thought a lot about, is it about the viewing distance or in the scanner? We ran an in-lab comparison with the instructions. I also meant to say that the behavioral relationships we didn't get for rest with memory scores. So it may be that memory is not sensitive enough in these cases, but I think to be continued, yes. And other people can get questions too.
Thanks for the presentation, Angela. My first question was really Cindy's question. I was waiting to look at the correlation, and I was expecting a reverse correlation in the sense greater hippocampal activation, greater fusiform gyrus activation. Is that related to better performance? So then, the question is, how does it impact memory? My second is, have you looked at, Asians who are in Asia versus Asians who are in America versus America, because culture is not just a static thing, it's dynamic, right? But at the same time, we know that microcultures in US do tend to hang out together. I still wear saris, and I still hang out with my Indians. I would not wear sari in front of you guys because I'd look like a freak. So there are, we tend to hang out together. And so I was curious about, have you looked at what are commonalities and what are the differences between the Asians in US versus,
Yeah, that's also a great question, and one of the places we want to head. So far it's really been comparisons across countries or people who moved to the US recently. And with this, we've done it multiple ways. So again, it's been nice to see the replication. We haven't studied Asian American samples, partially because I think we're going for the bigger difference, and then the goal is to get there. Is it an in-between group? But really, the heterogeneity of people's cultural experiences and having good individual difference measures of what would then predict a more Eastern or Western pattern. One of the places we're trying to go in collaboration with Audrey Duarte, who some of you know, is to really look more at acculturation. And it's kind of shocking how few cognitive studies are done looking at any of these individual difference measures or as people move to a new culture. So the first grant submission did not do well at NIH. So if any of you are here and reviewed it, please give me more ideas. But I think it's a really important direction that we've thought about framing in a way that I think will be more appealing. So hopefully that will be coming in the next couple of years as a starting point.
And not to keep picking on you, but it might really be that your findings are the most rich in like an age by culture interaction, right? And you haven't gotten there yet, so.
And hopefully that medial-temporal stuff at Retrieval will come out then too, that everyone gets the pattern separation.
Yeah.
So I'll have a question for Thad to get you off the hook. So Thad, one of the slides you showed was the Leventhal slide. And I know I've asked you this before, but I'm just wondering, has there been evidence linking dedifferentiation in a particular area of the brain and deficits in distinctive perception, or some sort of specificity that can count, that caches out behaviorally? Now I know there's the motor piece, but how about in sensory cortex, for example?
No. So we have, the sensory motor data is the one where we found a clear relationship. ironically, for the visual cortex, it actually relates to a more general. It's not just, I mean, some of the fluid measures involve, all of them, I think, involve vision, but it seems to affect all of the, I think we had 8 to 10 different fluid processing measures, and they were all related to, house face distinctiveness. So if people have hypotheses about why that is, I'd love to hear them, but I don't know the answer.
I don't know that Naftali knows the answer, but he wants the mic.
No, that's the last thing Naftali has is the answers. But I have a couple of questions. You took this distinctiveness measure from the studies in monkeys, right? Why did they need that measure as opposed to just talking about signal-to-noise ratio, which in aging sort of seems like a more parsimonious explanation with quite a bit of literature on it. So is there anything special about distinctiveness?
Good question. So we have actually looked at our measure, or one of the measures we've used is this within condition similarity minus between condition similarity. And so we've looked at actually those separately. And it actually looks like the within condition reliability, first of all, they both change with age. So both the within condition reliability declines with age and the between condition similarity goes up with age. The term, and maybe it's it was the wrong term to choose originally, but the term neural distinctiveness sort of seems to emphasize the between condition difference, but there's nothing in the data that says it's more important. And in fact, we don't have significant results, but when we look at the behavioral relationships, if anything, they look a little bit stronger for within than between. So I do think actually a lot of it has to do with reliability and signal-to-noise ratio. Now let's ask another speaker.
Well, my question was for you. I had prepared like a softball question to kind of warm us up, and then like a harder question. I can't for the life of me remember the softball question. So
bring it on.
So here we go. And it's not just for you. It's just what I'm interested in recently. So you've probably seen the recent literature looking at like inhibition and GABA in default mode, like deactivation. And so I thought that could start a lively, spirited debate, something about what are we tapping into. The GABAergic correlations with the deactivation are pretty surprisingly strong. And so I just kind of, this seems like a good piece for discussion.
Yeah. I'm less of an expert on the resting state stuff, so actually I would love to defer to others because I'm also very interested in that question. So if anybody that does the functional connectivity stuff has thoughts on that relationship, I'd love to hear them.
Or not even resting state, just the act of deactivating a system to hypothetically allow another system to rise up and do its job.
Yeah.
Seems very intuitively to be like an inhibition-excitation trade-off.
Agreed.
So of course you go
Agreed.
to GABAergic and glutamatergic mechanisms for that. So I'm thinking that it's this little cookie trail that's just too neat to follow, but
Yeah.
my brain intuitively wants to race down that trail because it is so intuitive.
I mean, the one thing I can say about that is there's a lot more spectroscopy work on GABA than there is on glutamate, but there is starting to be efforts. So it's harder to measure glutamate by itself, so it's usually measured as so-called GLX. And we have actually looked at GLX, and if anything, it's maybe better related to behavior than GABA is. So I do think considering both and the ratio of excitation and inhibition is critically important and is something we'll be doing in the future for sure.
Yeah. Naftali?
Nick.
Oh, Nick.
I wanted to say, I mean, I think it's entirely an empirical question as to how these things play out, but I don't think fMRI bold is a great way of looking at excitation versus inhibition ratio, because GABAergic synapses themselves are metabolically expensive. They generate increases in bold signal, and whether the net consequence of an inhibitory or an excitatory effect is a positive or a negative bold signal, I think is probably very context specific and probably regionally specific. And I seriously doubt, I don't know, Thad, what you think, I seriously doubt there's any general principles at the moment that we could adduce to make an inference about changes in inhibition excitation ratio from looking at fMRI bold.
I totally agree. And I was actually just talking about MR spectroscopy measures of GLX and GABA, and even
Sure.
there, you know, whether you can get a really pure measure of the ratio is.
I think
It's
there
done.
you get I think that's much safer ground.
Yeah, agreed.
Except you don't know how much of the GABA and the glutamate is in the cell.
Absolutely, yeah.
And how
So-
much of it is actually important for synaptic function as opposed to sitting around in extracellular space.
Yeah, that's exactly the point I wanted to make. And in particular, so one very promising technique that I think may begin to overcome that problem is paired pulse TMS. So we have started doing paired pulse TMS where you can actually, you're not just measuring, you know, I mean, I think about MR spectroscopy of GABA as, you know, you take this giant voxel, you stick it in a blender, and then you look at GABA levels in this huge area. The nice thing about paired pulse TMS, it's a functional measure of cortical inhibition, and so you can actually get it. So Dalia Kamash, who recently defended her dissertation, now has a measure of a paired pulse TMS measure of cortical inhibition in visual cortex, and she's found that it is highly correlated with the MRS measure, but this is a functional measure of real cortical inhibition, so I think that might be a very promising technique to use in the future.
I think I have the same thing to say as Naftali, so I'm going to let him say it. We'll see. We'll see.
No, just to continue this thread on glutamate and GABA, I think it's extremely important to do GABA and glutamate in the same subjects. And yes, you can see glutamate, not just GLX, and what you need is MRS whisperer and I happen to know a person who can do it. And they're actually, I mean, it is a very difficult thing to do on 3T, which brings, again, the question that we're not going to move forward until we have 7T spectroscopy, where we will be able to measure both metabolized smaller voxels, get rid of the white matter contribution and all this stuff. But one way that, and I have not seen this done with GABA, but with paradigm that Jeff Stanley developed, we're doing the functional MRS. And in functional MRS, of course, you, within the same subjects, track the change from encoding to, in the middle, we have just some tested blocks rehearsal to retrieval. It's messy, I mean, but you just need more subjects to sort of see clearer results, but it can be done. And I wonder if the same can be done with GABA, like literally within the memory cycle. So I don't know. Do you know of any GABA literature?
I asked Jeff that question,
Yeah.
actually, and to my knowledge, there isn't. I mean, there isn't good work doing it yet.
Yeah. Well, good luck.
It seems particularly good for aging research, though, because if you can leverage this glutamatergic functional imaging, you've wiped out these bold, neurovascular coupling problems. Maybe not wiped out, completely, very, very much mitigated that. So I think if it could get easier to do, I think it would be a really great way to move toward. Yeah. Yes.
I had a question for Michelle. That's why I stared you down first. No, I really enjoyed your talk, and one of the things I wondered, given the data set and the Experience Core project, is have you thought about any of the social factors or interaction pieces?
Yeah, I think in part, we always understood it was a social affiliative model, but we didn't have measures, so we actually included standard measures like social network size and how many people can you contact. But I think we need better, more thoughtful measures from self-report of social engagement. We also try to measure social activity, but that's hard to do because you're looking for the primary driver of an activity, and it's a judgment call whether someone thinks that going to the movies is a social activity versus a more solo activity. So it's a challenge. Our answer has been to try to incorporate GPS into wearables to think about whether or not people are going to places that naturally, by definition, are social, like going to the grocery store, where we're going to assume that's a social activity, or going to the park or something where we can compare their physical activity on the accelerometry with their location. I know it sounds a little bit stalkerish, but we are de-identifying the data. So we're looking at heat maps to look at patterns of data based on Google Maps. So that's our approach to look at social engagement. But within an intervention design, I think you just have to a priori establish how you think it's going to occur. And in Experience Core, it was pretty easy to see. And in the shark game, which is actually Bandit, I'm going to say sharks don't get the primary ticket there. It's really about Bandit, yeah. And the dolphin is just so socially affiliative, and it draws empathy. That I think that's something that is engendered my interest in the amygdala and the socio-emotional network, which I don't know that much about. And I'd love to learn more from people here about how to measure the amygdala or the networks around it.
One more?
Oh.
One more?
I just had a follow-up for that question to Michelle. So one question was about the survey that you had, that I really liked those 7 questions. And one of them was about church volunteering. What about people like few of us who are atheists or don't go to churches or were never exposed to it? So can you think about how would we define, some of those variables into a non-majority population? And the second one was, I was really interested in the variability in the learning rate. So I was thinking if you have day-to-day some kind of measure that could look at how the learning rates could be influenced by these, things like sleep or mood or, variables that could potentially predict variability in how you're learning, how to save the bandit from the sharks.
I'm going to ask you to answer the atheist question. I don't know if you're agnostic or atheist, if that's like a negative or a positive, and I'm joking, but I don't know what we're looking for are activities that we know people will remember. So if we can think of other activities when you're young that you just remember doing or regularly, we would love more activities. So so it's really it's just really a lack of creativity on our part to think about what that activity might be. I have to say, I can fully appreciate your question from many points of view. With regard to the other question about learning or performance, I think broadly speaking, there are so many performance metrics that we could get that might teach us about strategy, for example, that I didn't talk about today. So when people are learning, all I can say right now is that our first question was, could everybody learn? And because we had so many varying levels of ability to start with, could we get everyone to get better? And I think what was amazing is that everyone did. And some of them had mini mentals of like 15. So they were cognitively at risk, but we showed that even they could learn. They didn't show the same steep learning slope, but it's encouraging to think that most people could learn. I don't know about environmental or contextual factors, like if they had a cup of coffee. It would be interesting to see what contextual factors.
You have it in your survey or something.
Okay. Well, in 10 minutes, we're looking forward to looking at Mick, looking forward and looking backward. And whoever picked this picture, kudos, man. That's the perfect picture for that talk title. So in 10 minutes, we get to look forward to this.